Welcome to our Grand Rounds series. Our Greenberg bladder cancer team are grateful you're here. Of course, seeing you in person would be preferable, but these virtual platforms have proven to be very efficient for sharing information, for education, and most importantly, for discussion. So we've got a couple of updates for you. Where our new Grand Rounds session is online. You'll see if you go to Greenberg Bladder Cancer Institute, you'll find a complete list of them there. And I wanted to also draw your attention to our evolving group of programs specifically tailored around the experiences of women with bladder cancer, developed and led by Drs. Armin Smith and Jeannie Hoffman-Census and the program team of Pamela Getz, Samantha Rockler, and our own Stephanie Cooper Greenberg. Again, the programming details are available on our website, and of course, never hesitate to reach out to us, and we'll send you the details directly. Just email us at bladdercancer@jhmi.edu. So we have an unusual situation today where I'm the host, but I'm also the guest. So just to introduce myself, I'm the director of the Greenberg Bladder Cancer Institute. I went to college at Brown University, and then I did my PhD at a place called the Karolinska Institute in Stockholm, Sweden. My major there was biochemical toxicology, which is kind of relevant to what I'm doing now with Sunil Patel. But I actually did a postdoctoral fellowship at Dana-Farber Cancer Institute in immunobiology. And again, if you're around long enough, I guess whatever you do comes back again. So now, as you all know, we're doing a lot of immunobiology and bladder cancer, and it's thrilling to be part of this new wave of excitement around cancer immunotherapy. I spent a long time at MD Anderson Cancer Center, where I ended up being the director of urological research there before I joined the GBCI in 2016. And I'm really thrilled to be here and to have as our host, our own Stephanie Cooper Greenberg. Stephanie, I'll turn it over to you.
Okay, David, get ready to be tortured for the next hour.
Happy to.
David, you've been at the Bladder Cancer Institute for almost 10 years. It's amazing how time flies, and hopefully a lot of progress has happened. But I think the intent today is to share with everyone how it really is a golden age for bladder cancer research. There's so much that has happened in the last, to call it 10 years. Things that did not exist when this institute was created. There was really nothing. There were very few clinical trials. There was very meager research. There was hardly any sequencing. There was hardly any idea of really what is going on with this bladder cancer and upper tract cancers. And it's just exploded. I can say that with pride, that the research has become robust and serious and important and has been transformational as it's helped patients, which is the only reason we're here, is to improve the patient experience, improve longevity, improve outcomes. This is what we try and live for every single day. So I wanted to ask David just to explain a little, it's not like, making legislation in Congress. We're not going to do that to you. But I have found there's always a sense of awe and wonder when you hang out with David McConkey and our fellow faculty members who have full-time jobs being surgeons and oncologists, and who happen to do research on the side. And also our young lab workers and PhD people and postdocs, and these remarkable brain trust of passion and drive to sort of find the clues to find cures. And I'm not saying that word lightly, for bladder and upper tract cancers. So the first thing I wanted to ask David, and this is a rambling conversation, please ask your questions. This is the moment to ask, like, how does this work? Or what's with that? I ask these questions every single day. Our goal is to keep it in English, so I interrupt my dear friend David from speaking science too much, so that we can all understand. And also, hopefully, you'll walk away feeling that actually a lot is being done that's very positive. A lot is being done that's immediately helpful to patients, and that there's a lot more to do, and that they've really kind of come a long way. So, but without, I've been made aware that without the patient's involvement, there would be no research. So David, my first question to you is, how do patients um, uh, make research happen? And this is just a general question. How do they contribute to the research process? And you can speak in specifics about what happens at Johns Hopkins on a daily basis, as an example.
Why don't I start there? Yeah. So here at Johns Hopkins, we are asking our patients to consent to be part of our research. And we're asking as many of our patients as possible to be part of our research. And what we do is we present them with consent, which maybe some of you on the meeting have received already, that requests that we use their clinical data and their tissue to perform experiments, both now, experiments that we know we want to do, and also to reserve the right to continue to do experiments in the future with the same data and the same tissue. And so, honestly, the number one priority for us is to get the kind of outstanding quality clinical data that we get when we enroll patients in clinical trials. So the best possible clinical data you can get are the clinical data that you collect intentionally in advance, rather than going back into the chart later and looking back and seeing what's there. So we have a team in the Brady and the GBCI who go to the OR, consent the patients there. Sometimes we, do a remote consent by telephone, but we prefer not to do that because we prefer to talk to our patients about what we're doing so that they understand the importance of what they're signing up for, and also so that they can get as excited about it as we are. And then we do not take tissue that needs to be used for clinical diagnostic purposes. We're very careful only to take any extra tissue that might be left over. So we focus most of our measurements on what we call formalin-fixed paraffin-embedded tissue, which is what the Department of Pathology does when they cut a tumor out of a patient. They put it in what we call a formalin block, and then it's stored there for as long as the patient needs it, which is in many cases now, with patients fortunately living longer indefinitely. So we have to fill out a protocol. It has to be approved by the IRB, and then if the patient consents to our research, then we can get the tissue we need mostly through those paraffin blocks that are being kept in pathology. The blocks never leave pathology. We'll get slides cut off of the block that get sent to the laboratory for investigation. But again, it's the number one priority is to make sure that the clinical care of the patient is optimal. And then in addition to that, we'd also like to use the clinical information in the tissue to understand the biology of the disease.
So for the patient, it's whatever they're removing would be removed anyway and maybe thrown away. It's not an extra step or anything for the patient. It's just valuable tumor information, and that tumor information, if I have it right, then you do genetic analyses on the tumor.
Right. Well, in some cases we are asking for something extra, but usually it's either blood or urine.
Oh, well, we have plenty of that.
Yeah, we'll only get the blood if the phlebotomist is taking the blood for diagnostic purposes anyway. And then with the urine, that's actually our most exciting fluid right now. Hopefully that isn't too hard to part
Give it to
with.
bladder cancer to say urine is exciting. Thank you, David.
Yeah.
Always turning it into a positive. Well, it's sort of like a might as well. You might as well, be part of research and look what it can yield. That's really fantastic. But for the patient, would you say it's a burdensome to be part of research?
No, I think the biggest concern I've heard most patients speak is that they're worried about, privacy and what might happen if, the results of our sequencing data escaped and made it out into the real world. And we're very careful to de-identify everything and maintain custody of everything. There's really, in my experience, and I've been doing this for a long time, there's never been a breach. You hear about these data breaches where Home Depot loses your credit card account numbers or something. We've never had one of those when we've been working on this research. So
you work hard to make sure that won't
absolutely, and the IRB helps us do the right thing too.
IRB is internal. What is that called?
Institutional Review Board. That's
There's a lot of people overseeing this process.
yeah, yes.
And
Yep.
this is common for not just bladder cancer, but it'd be for other cancers as well, other solid tumors. It's the same sort of,
That's right.
all right?
So here at Hopkins, we have these research entities called Precision Medicine Centers of Excellence. And we're all doing the same thing. We're all collecting clinical data from our patients in an intentional way so that the data are high quality. Because that's really, in the end, as they say, garbage in, garbage out. We can do the best sequencing in the world, but we don't have good clinical data to go with the sequencing data. It's just junk.
Okay. Well, let's move on. So thank you for that. And to all patients who consent to research, we thank you because
Thank you.
you're paying it forward and the research is a long journey, as we all know. We wish it were more, which were quicker. Some days, sometimes it is, but without the patient involvement, there would be no research and no
That's right.
progress. So thank
That's
you
right.
all to those of you who consent and are part of the process. We thank you for that. Okay, first question. There's so many different kinds of research that we kind of hear about as lay people. Can you explain what is translational research? What does that really mean?
Well, simply put, it's translating things from the bench to the bedside or the bedside to the bench.
Okay.
So it's research that's focused on solving a human disease problem. So there's also basic research, which is really focused primarily on understanding a basic biological process. And a lot of our most important discoveries have come from experiments that were done in yeast or fruit flies or nematode worms. Really, really important basic discovery research on very basic biological processes. That's really critical research that needs to be done. But here in the GBCI, we're definitely more focused on translational and clinical research. We have adopted an approach that I think is very effective, which is we like to do our discoveries in people. And based on what we find in human disease, we bring it back into our laboratory to try to understand it using model systems that are easier for us to manipulate experimentally. So it's almost always coming from a patient and coming backward to the lab, as opposed to, for example, I have a lot of colleagues who've made discoveries in mice, and then they've translated those discoveries into people. That's not really the way we prefer to do research here at GBCI. We prefer to do it the other way around. Hence the need for everybody who wants to participate in our research program.
So would you say that research is about, having curiosity and seeing patterns or observing as a clinician or the urologist or the oncologist, they see something and they maybe see it a few times or in one remarkable patient and then say, well, why is that happening? Is that where the root of all research, is that where it's coming from?
It's where a lot of the best research comes from. So to give you an example, and we're all very proud of this, our own Jeannie Hoffman census, working together with a talented postdoctoral fellow, noticed that in patients who were getting benefit from a drug we'll probably talk about later called enfortumab vedotin, they developed skin rashes. And so Jeannie was wondering why there was this connection between getting a skin rash and having benefit from this drug. And so first she did a very thorough job of counting how many patients got the skin rashes, how they turned out, what their outcomes were with this drug. And she confirmed using, rigid statistical approaches that there was in fact a strong relationship between the two. And then we need to understand why. We need to understand the mechanism. And so we were aware of other research going on here at Hopkins. It was being led by Anthony Rosen and his wife, Livia Cassiola Rosen, where they had noticed that patients sometimes have an autoimmune skin disease that presents right before cancer. And it doesn't have to be a bladder cancer, it can be any cancer. And it can also work the other way around, where a person can get cancer and then have a skin autoimmune disease. And so because we're in an environment, where everybody is collaborative and talks about these things, we knew about this work. And so Jeannie and Alina have partnered with Anthony Rosen and his wife Livia to perform mechanistic studies. Again, this is backward from the bedside to the bench to understand the mechanism, and the preliminary data look astoundingly positive. So
if you're the patient,
mhm,
thank you, patient, and you get this drug, which you'll talk
mhm,
about because it's fairly new, and
mhm,
you have this awful rash, which is uncomfortable and miserable, but you're doing well,
mhm.
something's happening, meaning the drug is maybe causing the rash, but it's maybe
Yeah,
causing something good.
yeah.
It's like when we had the COVID shots, right? We always got the COVID shot. We kind of wanted to get sick, like maybe it's really working, it's just a stupid psychology on my part. I wish that it didn't happen. But so something's going on, in other words. So the question is, what the heck is going on? And so somehow they find patterns of people similar who've had the rash and a good
Mhm,
outcome.
mhm.
And so that means this is a good drug for
Means it's a
certain.
good drug. And a more kind of typical way we do this is, Noah or Max or Jeannie will be doing a clinical trial, and we will collect tissue and urine and blood from the patients enrolled in that trial. And we'll do all kinds of measurements, we'll sequence stuff, and then we'll see how the patients did. Some of them will have done well, some of them maybe not so well, but then we'll correlate the outcomes with all these measurements. And usually what we see is a pattern. We see there's some pattern in the genomic data or some pattern in something else we're measuring that correlates either with sensitivity to the drug or resistance to the drug. And once we see that pattern, then we take it back to the laboratory to see if we can establish cause effect. We wanted
And
them
can
to
you study
understand that.
all the genetic profile of these patients too, like
Yeah.
what's going on
So,
genetically?
right. So just to be clear, we have established a panel of 60 genes that is mutated in patients with upper tract and bladder cancer. So we've really pulled out the top 60. And we have a sequencing assay that we do right in the lab.
What does that mean?
So,
What's a sequencing assay?
well, so we
You
need to
give a
find the
concept word,
mutations.
but we
Yeah.
don't know what that means.
So we need to find out what mutations are there. So we take the tumor tissue and we grind it up, and we use various chemicals to pull the DNA out.
Huh.
And then we sequence that DNA on the machines that are sitting in the lab next to me.
What does the machine look like?
It looks like a box, a plastic box that sits on the lab bench. But if
Oh,
anybody wants to come over and look at it, you're welcome to. But
you
it
can
has a
have a
lot of,
sequencing machine on a desk, like a big
yes,
printer or something.
yes. And we've got a couple of big computer servers where all the data are stored. We also
Really?
have backup data storage across campus here.
What
But
comes out? Is it like a report, that looks
yeah,
like a stock chart or something? Is that what
yeah, it's all electronic. We've got certain formats of data that come out right off the machine. The data go up into the cloud, and through data science, the sequencing data are made into mutations and other things that are significant cancer.
So it would show on the panel of 60 genes that you have
Mhm,
selected, the
mhm.
flavor of the day,
Yep,
it would show something's going on in some of those genes of the
right,
six, and it's telling you something.
correct. And we then are using those results for our research. In parallel, Noah and Jeannie and Max and the
Wait, let me
others.
stop you
Yeah,
there. Don't go,
sure,
don't leave me yet.
sure.
This is where treat me like a 2-year-old. So if it's shown that you have whatever, is the point of this that someday you can be predictive, like, oh, Mr. Jones or Miss Smith, if you have this and this kind of tumor and this kind of rash, and we gave you this drug, this is the drug for you. And then you can tell other people with a similar profile that this may be the right drug for you moving forward.
Yeah, so we've gotten pretty sophisticated these days. So back when I joined the GBCI in 2016, I was working together with colleagues at Fox Chase, Betsy Plimak, and colleagues at Memorial Sloan Kettering, Jonathan Rosenberg. And they had developed a very interesting observation, again, based on clinical trials, that patients who had certain mutations were responding better to chemotherapy.
Okay.
So we've done a ton of follow-up validation work on that, and the connection still stands. If you have certain mutations, you're more likely to respond to chemotherapy, but it's not black and white. If I have a patient standing in front of me and I find one of these mutations, I can't say with 100% certainty, you are going to benefit from chemotherapy. So now we're adding all kinds of other measurements to that. We're adding urine liquid biopsies, we're adding plasma liquid biopsies, we're adding microbiome, we're even adding something called artificial intelligence pathology, or AI path, which is where we literally take pictures of the slides the pathologist uses to stage and grade a tumor, and let the computer figure out what's going on based on what the computer sees on the slide. So we actually have a lot going on.
So when you started 10 years ago, were there
Mhm.
was sequencing being done?
Sequencing was being done sporadically. So our friends at Memorial were really forward thinking. A colleague there named David Solid put
Yeah.
together one of these protocols, these clinical protocols I was talking about that need to be approved by the IRB, where they essentially enrolled everybody at Memorial Sloan Kettering to the protocol and sequenced everybody.
That's a lot.
And so that they have thousands and thousands and thousands of
But
patients.
sequencing itself, is it the last 10 years that it's really come to be a thing? Or like
Yeah.
it's like a standard of research, right? Is
Right.
that
So now a lot of patients, maybe some on this meeting, will have their tumors sequenced by places like Foundation Medicine or Tempus or Caris, or maybe even by the molecular lab here at Hopkins. And that's a clinical test that's used for clinical decision making. It's a lot more expensive than the one that we do here. So ours is generating the same quality data, but it's not being used for clinical purposes. This is used purely for our own research.
But do you see this work,
Mhm.
just the example of the skin rash and this drug, and then throwing in all the other things that are, we'll be talking about biomarkers and whatever. Do you see this as a day will come when this will be largely predictive? Like,
Yes,
you'll be able to say, hey, this is going to work for you, or this is not going to work for you. I think
that
one of
is
the most
absolutely the
important
goal.
things to know is if a drug is not going to work for someone,
Exactly.
like chemotherapy, why suffer? And it is true suffering to go through that if you know in advance it's not going to work. So that's the
Right.
beauty.
Right.
Wow.
That's really the overall priority in all of it, is to figure out who's benefiting from therapy and who's not. And when I say therapy, I mean everything you can think of,
Right.
surgery.
Are we going to get there, David?
Yeah, we're going to get there. It's
Okay.
going crazy fast.
Okay, I'm holding you to that.
Mhm.
Here's Dr. Armin Smith, our beloved urologist based out of Sibley, has been fascinated recently and probably throughout her whole career, but has started some survey research that I want you to
Mhm.
talk about. This is now we're
Sure.
flipping gears here from the bench to bedside in the lab and the um the clinic to the lab, or whatever way it went. So Armin again is remarkably compassionate. She talks to her patients, and she wants to hear from her patients. And so can you explain what the quote unquote patient reported research that Armin is doing as an example, and what that is in the
Sure.
whole lexicon of like research?
Sure.
So it has the patient voice in it, and how is that used?
That's the other thing that's happened that's really exciting over the last 10 years, is that there's a lot more focus on what patients want and how patients are feeling, how patients are thinking about things. And again, it's been because people like the ones on this meeting are willing to be participants in a lot of this discussion. I know you've served on the task force. I know that our friend Rick Bangs has been heavily involved.
Mhm.
There are a lot of people like that who've spent a lot of time being advocates for the whole community. So patient reported outcomes are outcomes that are usually obtained by surveys. So a patient will be asked to take surveys, and based on the survey results, the research team will correlate the results of the surveys with some other clinical outcome of interest. So
Is that for like quality of life and maybe best
yes,
practices? So
yes,
the two specific studies, which our beloved Armin is doing, which are remarkable, is a survey for women about sexual function and wellness and the challenges of, um, you know, radical cystectomy in women, and a lot is involved with that, and also sexual function for men and women. I think she's doing a study right now specifically for women, asking women to fill this survey out and respond to questions so that perhaps it can inform a way to ask the better questions. Surgeons, physicians, I think always try to ask good questions, but sometimes that gets lost and the patients feel lost. So she's doing these surveys that will hopefully end up in a paper or in a conversation where people will know you can ask these questions because this is what people want to talk about.
exactly, exactly. And mood is another example, right? So we want to know how people are feeling throughout the course of being treated themselves and also their care partners.
Yeah.
There's a lot of emphasis on patient-reported outcome.
So those studies are at the national meetings where they would be presenting the findings of, X number of women feel that, they were never asked the question, or it wasn't really, they weren't really fully informed, or it was never even brought up, which happens quite a lot. And then it would just maybe inform best practices in terms of
Exactly.
the right questions to ask, but it would
Yeah.
be elevated to sort of a standard of care.
Right.
Is that correct?
That's correct. And everybody's taking these very seriously. So, I hope everybody realizes just how crucial this is for our overall research program
Right.
to have patients be willing to participate in, because it does take extra time.
Yeah. I will add that, and you know this, David, and please pipe in that, Armida is also she's doing a sexual function study in women. So asking women, just basic series of questions. I don't think they're terribly long. Anyone can participate, by the way. And Armin the study is open. It's just surveys, and then they compile the information, data, people figure out what the percentages are, and then it gets written up. But the second study I found really fascinating is working with psychiatrist colleagues, whose name escapes me. There has been anecdotal and observational evidence that BCG may be causal for depression, which is something I had never heard of before, because there's a certain gene expressed, help me, David, IL-6.
IL-6, yeah.
And that they're seeking out patients who have received BCG and have also, as a causal factor, are sort of afflicted by depression. Now, they don't know if it's, of course, bladder cancer is a thing to be depressed about, but they don't know if it's causal because of IL-6, which is what, a protein? What is that?
It is. It's an immune protein that tends to swim around in your blood and certain other places.
So the
Yeah.
BCG can trigger IL-6?
So BCG is confined to the bladder.
Yes.
So it's not like the IL-6 is coming directly from BCG. It must be happening through an indirect effect somehow.
So she's working with a psychiatrist. It's a study that they've been just started, and again, this is a survey. Did you have this happen to you? And then they can learn more. So
Right.
that is patient-reported outcome research.
Yeah.
And thankfully, it's becoming more standard in bladder cancer, and it didn't seem to take on, it took a while to get here. Is that right? Would you say?
I think it did. I think it took too long. I think, though, doctors are starting to actually change their practices based on the results of these studies.
Good.
So another example is organ preservation in women. It used to be that women getting cystectomies had a lot of other stuff cut out, and Armin is pioneering an effort to change that too. So
Yeah.
there are a lot of examples of how now patients are telling doctors more what they want, and doctors are listening to them because of it.
If any of you are interested in our mean studies, which I did not do any justice to, we will connect you and just email us at bladdercancer@jhmi.edu and we'll connect you to our means. She's working with a really fantastic research assistant, and that work is ongoing, and it's really, really fascinating. So it's, it's about treating the whole person after all. Okay, now we're going to switch gears. Sadly, we learned this week from the American Cancer Society that colorectal cancers, for some reason, are getting more prevalent in young people under 50. That was not the case for the trend for years and years. And again, we're talking about colorectal cancers. Anecdotally, also, it's been shown that bladder cancer cases are trending to younger people. And again, no one knows why, but smoking, of course, is causal for bladder cancer. Everybody knows that, and it's secondhand smoke is implicated in that too. But what can you talk about, David, about epidemiological research and
Sure.
exposure to chemicals, and how, if will there be a moment where you can tell causality with
Yes.
pollution, environmental factors,
Right.
chemical, pesticides, lawn for PFAS is a big one in the military and firefighters. The plastic bottles that don't have something, some something, microplastics. Yikes! How do you do research on the environment related to bladder cancer? How do you do that? And wow, how do you figure it out?
It's complicated, but we finally are developing the tools to do it really well. So for those of you who read The New Yorker, I think it was one of the issues in December, and I'll see if we can't put it up on our website if we
Great.
can't at least provide you with a link. Sid Mukherjee wrote an article about a paper that one of our colleagues published. His name is Charles Swanton. He's based in the UK, where the conclusion was that air pollution does promote lung cancer, but it does so through a mysterious mechanism. So we thought that the particles and other chemicals in air pollution were causing mutations, were damaging the DNA of lung cells. And lots of studies had been done to try to see if this is true, and they were, for the most part, negative. But now we understand that there's a second way that chemicals can be harmful, and that is if they happen to interact with a cell that already has a mutation, then it can blow up and turn into cancer. And a lot of the mechanisms involved in this effect, which we call promotion, tumor promotion, involve inflammation. So if you can think of all the different ways we've talked about people being exposed to inflammation, there are also all kinds of different ways that this must happen. And it's a combination of having the inflammation and having the bad luck to have a cell that's just perfectly suited to respond to it. So it's not like anybody who has inflammation is destined to get cancer. But now that we're starting to appreciate that people sometimes just spontaneously develop mutations in their bladders, just as they do in their skin or their lungs. This just happens as we age. And we don't necessarily need a carcinogen to have that happen. So people have worried for a long time about what they might have done wrong to develop bladder cancer and other malignancies. And in the end, maybe a lot of the time, you didn't do anything wrong. It was just the accident of age and the mistakes in our DNA. But we can now start to see, in some cases, mutation patterns that are extremely well tightly correlated with certain carcinogens. And so that's a smoking gun. And then we can also develop, again, laboratory models, which we're doing in collaboration with Dr. Patel here, where we make the mouse have one mutation that we know is important in bladder cancer. And then we expose the mouse to all these different environmental chemicals that might be carcinogens. And we ask what happens. And in some cases, I'm sure we're going to find that these chemicals in the environment act as cancer promoters. And then we can start to think about how we can prevent people from being exposed to them.
So does most of the epidemiological research happen in mice, would you say?
No, that's the really hardcore laboratory stuff, but most of the epidemiological research happens in large populations of people. So the first clue that something might be going on is, for example, there are increased cancer rates in people who are exposed to air pollution, or case of bladder cancer, there are,
Well,
a
our
number of different
beloved
things.
veterans are, and Agent Orange is a
Exactly.
couple.
That's
Yeah.
going to be number one experiment, is to see whether Agent Orange acts as one of these cancer promoters. So,
The Agent
but
Orange was Vietnam War. I mean, it
yeah,
was,
it is. But the chemicals that are similar to Agent Orange are found throughout society. Just because Agent Orange isn't around anymore, and the reason it isn't is that the chemical's super stable. It was stable enough to survive, long periods of time in Superfund sites. I actually studied it as a graduate student when I was in Sweden.
you
But
did?
yeah, I did. Dioxins is
Did you
one of my
get
first papers.
exposed, or do we have to worry about
I
you?
was
Or
wearing a NASA spacesuit. I think I was probably more careful.
were you careful?
Yeah, I think I was probably more careful than other people running around on the streets being exposed to dioxin-like chemicals on the streets. But
It's like a horror movie.
yeah,
Wow.
no, the epidemiologists, and we're fortunate to be working with Elizabeth Platts here at Hopkins, who's fantastic. They show us the patterns in large populations of people. So the patterns would be people living in certain areas who might be exposed to certain things or developing cancer.
Do they look at occupational causals?
Absolutely.
Firefighters and
Yeah.
PFAS is a thing, right?
Yeah. Yeah.
Many other.
They collect as much information as they can. And just so we were talking about at the beginning about clinical data, it helps if it's collected intentionally.
Oh,
So
okay.
there have been some very large prevention trials that Elizabeth's been involved with that have collected intentionally in advance all kinds of clinical data, including occupational exposure histories and so forth. And we can mine those data sets because sometimes they were designed to study heart disease or prostate cancer. The prostate cancer trials, it turns out, is revealing something about radiation exposure in bladder cancer. So we're working together with Elizabeth and some colleagues of mine in the Southwest Oncology Group to
So
understand.
just to circle back, and I have a great
Yeah.
question, Karen, about what we are just talking about. When a patient is part of the research process and they agree to donate and into the biolab, the library of specimen library, or for lack of a better word, their contribution could be studying the skin rash thing or the PFAS environmental stuff. I mean, it can be used in a lot of different ways for a lot of different kinds of research. Is that correct?
Absolutely. So
Oh,
by
that's beautiful.
creating this database, we expect that people are going to bring all kinds of different questions to it, and because we have it, they'll find answers.
Okay,
We
now
did this
the
did
housewife
this
is
intentionally. Yeah,
stored in like refrigerators or like in a underground, or what the
that's actually a big problem.
oh,
So storage is a big deal. So
generators, please? Yes.
yeah, no, it's a big
Oh,
deal, not just for
fascinating.
us, but everywhere. So yes, we have big refrigerators. We have off-campus big storage areas that have tanks of what we call liquid nitrogen, which is super cold. We also, as we have developed more and more sequencing data, we're digitizing everything. So we'll get the DNA and the RNA sequencing data and put it in the bank, so we have that for future use. And then, unfortunately, we can't really predict. Technology is moving so fast. We can't really predict what kind of tissue is going to be compatible with the technology 5 or 6 years from now. So we just do our best. And we bank very intentionally. We have a review article if anybody wants to read it. We just generated with a whole group of investigators across the country about tissue banking. If anybody wants to read it, I'm happy to share it. So
I can't wait.
yeah.
So I have Karen has asked us a question. I'll just read it directly. Research protocols through COG, COG, I don't know what that is, have incorporated patient-reported outcomes into medication trials. Do
Mhm.
you see that happening with adult trials?
It's happening. Yes,
What is
absolutely.
COG, by the way? I'm sorry.
Children's Oncology Group.
Oh, okay.
Yeah, yeah, that's one of the things we were going to talk about was
Well, that's my
cooperative
next
review.
question.
Yeah,
So
yeah,
nice segue.
yeah.
So so the answer is
Yes,
patient-reported outcomes are now
yes,
incorporated into adult.
absolutely. So Sarah Sutka, who's at the Fred Hutchinson Cancer Research Center in Seattle,
Are
it,
answering my next question. You seem to know everybody, David, and you
she
as if
is
they're our
fantastic.
best friends.
Yeah, yeah.
Talk to us about research collaboration
Mhm.
and academic medical centers and other entities who do call it bladder cancer research and the collaborative nature of it. And also explain to us about the cooperative groups.
Sure.
How does that work? And the reason I'm asking it is I sort of know the answer, and I think people will be very pleased to know that it's a very collaborative
Yes,
enterprise.
yes.
Unlike maybe some other, territorial, academic pursuits. But
No, it's
you say it's
true.
collaborative and collegial.
It's always been that way. We've developed a very collaborative research community. I think in large part because of Beacon and their leadership in bringing us all together for the think tank every year. And we've maintained it, even though the field has gotten a lot more competitive. There are a lot more people doing work, which is great. We still work extremely well together. So,
Explain the cooperative groups. What is
sure.
that?
So nationally, the National Cancer Institute and CTEP, which is Clinical Therapeutics Evaluation Program, Cancer Therapeutics Evaluation Program, they run the cooperative groups, which are essentially collaborations of academic centers across North America and even Canada.
Mhm.
And they're given funding from the National Cancer Institute to run mostly large phase 2 and phase 3 clinical trials. Because with having these many investigators involved, you can get lots and lots of patients enrolled into these trials. These are big, big trials. So
Called
Max,
multi-center trials.
yes,
Right?
Max's Bridge trial is an example of that. So,
Explain
you
the
know,
bridge trial, decode that.
that's that's a trial that Max Cates, our own Max Cates, is leading. Phase 3 trial to compare BCG to intravesical chemotherapy with gemcitabine plus docetaxel. All the urologists love this trial. It's open across the cooperative groups, and tons of patients are going to be enrolled in that trial. And another example was the PRIME trial that was led by Rob Svatek, where we were comparing 2 different forms of BCG. These trials, they have like almost 1,000 patients oftentimes,
Okay.
and
So
you can't do these at home.
So
So
one trial needs a lot of critical mass.
correct,
So
correct.
they're shared among these cooperative groups so that
Yes.
they host these trials and then
Hmm.
enroll their patients.
Right.
And they all work together, right? Is it the same
They
criteria?
do. Yeah, we've got several of them. So, Max and Noah are members of ECOG.
What
Actually,
is ECOG?
Eastern Cooperative Group, on
You
Eastern
knew I was
Cooperative
going
Group.
to ask
Yeah,
that,
yeah. And then I'm still a member of SWOG, which
which
is the
is,
Southwest Oncology Group.
okay.
And so together,
Geographic areas in the country? Is that
it's
how it's spelled?
crazy. Yeah, no, it's not really. So it's more history than geography. So SWOG includes places like Columbia University, it's Yale University. So
Okay.
it's East Coast as well as Southwest. But
Okay.
we work together. So especially with our biomarker studies, everybody's involved. We've got everybody on these calls to design experiments using the tissues that we're collecting in these trials, which,
So, okay, that leads
sure.
me to a question. So we've just gone through the tutorial about the refrigerators and our beloved patients consenting and giving their
Mhm.
specimens for our library. Does that also happen at, say, Sloan Kettering and at MD Anderson? Is the same process everywhere else where their samples elsewhere, that everyone's doing the same research at the same time?
So we're coordinating that better than we used to. So
Mhm.
there's the kind of the barriers created by privacy, which are really important. So
Absolutely.
we have to develop strategies that allow us to collect the clinical data and de-identify the clinical data before we share it with anybody. So we use a database called REDCap, which is the industry standard, and it's built to be PHI compliant. It's built to be very easy to share
Privacy.
data,
Okay,
protected health information. Yeah.
thank you.
So we're loading all of our clinical data in REDCap with the expectation that we can de-identify those data and share the data with other people. And likewise with sequencing data, it's the same thing. We de-identify everything before we share, even before we publish. So that,
Is the idea that the more critical mass you have, the more data you have regarding a specific thing you're studying,
yes,
more patterns you can see or
yes,
validation, you need numbers here, you need critical mass.
that's right.
So for a study, for example, um, like the Genie study with the skin toxicity,
Mhm.
that may be a handful of patients, but someday it could be hundreds and hundreds or thousands. I mean, what kind of numbers
That's
are we?
correct. Well, she's already talking to her colleagues elsewhere about this work. They've made a similar observation now at Stanford.
Oh, okay.
And so the clinical data, correlating clinical responses with skin rashes, that's fairly straightforward. Where it gets trickier is sharing samples to confirm the mechanism. So uh, right now, we're collecting samples and analyzing them to see exactly what we think is going on there. Once we do, we can advise everybody what kinds of tissues they'll need to store in order to help us confirm the observation.
Okay.
But the
Got it.
clinical data we can share already, and that's what's going on. So
I have another question. Always wondered about this. What role do the pharmaceutical companies play in research? Do they fund you? Do they give you free drugs? You're also developing drugs too. So what role does pharma play? And are they good guys or bad guys?
oh, they're both, but mostly, again, they're good guys. They're getting the drugs to patients very aggressively. If it weren't for pharma, we wouldn't have so much activity going on in
When you
bladder
have
cancer.
David, I'm going to interrupt you, as I
Sure.
always, when you have a clinical trial and you're trying like a cocktail of immunotherapy drugs, for example,
Hmm.
Pembro and whatever, do the drug companies supply the drug for free?
Usually, yeah.
Oh,
So with these investigational agents, they're super expensive.
yeah.
So it's very unusual for an investigator to pay for their own drug or raise money for the
These
drug.
immunotherapy drugs are really costly.
Super
I mean,
expensive.
they're,
Yeah.
you
Yeah.
know, think
Yeah.
hopefully.
So industry runs their own trials,
So what does that
and
mean?
it means they organize them, they decide who's involved, they identify principal investigator, they manage the clinical protocol very carefully. Um, this is a business,
So
right?
would a
So
patient at Hopkins, a hypothetical patient, consent to being part of a pharmaceutical trial?
they would be told they're being consented to be part of a pharmaceutical trial. But a lot of our investigator-initiated trials are
Okay.
pharmaceutical trials.
Mm.
So Jeannie, for example, was developing a trial to study EV plus Pembro to see if she could allow patients with upper tract cancers to keep their kidneys. Now it's become standard of care. So now her trial has stopped being a pharmaceutical trial and looks like it's going to be a Hopkins trial. But in general, the companies supply the drugs and work with the principal investigator to design the protocol.
Got it.
Yeah.
I have to ask this question. You do a lot of research. My count is, there's about 20 projects going on at once.
Mhm.
And I've been told the projects never really end. They just kind of pivot
They
and
evolve. Yeah.
change and evolve. How is the research? This is not a softball question. I'm just curious. How do
Sure.
you pay for this? How do you pay for the chemicals and the mice? I guess you use mice
We use
sometimes. You live
mice,
in
yeah.
another building, is that true?
They live in another building. They're mostly Rusty's fault. So Rusty's the cause of most mouse misery, but
Not a big mouse girl, but
yeah.
is it, tell us about the grants and philanthropy and how does this, how do you get to pay for this stuff?
Any way we can. So here at Hopkins, we benefit from having generous donors. So yourself included. So we usually need some seed funding to get preliminary data. The way that our grant system works, it's difficult to get any kind of grant without any preliminary data. There are some grants where you don't need any at all,
Okay.
but they're very, very few and far between.
You need like spark funding and then you leverage that and get like an NIH grant or
Yes. Yeah.
uh,
Yeah.
do the pharmaceutical companies give grants?
They will supply sponsored research agreements, right? Not as much anymore with laboratory work, although we've benefited a little bit from money from Faring for the It's Children work that we've done.
Talk
But
about the Defense Department. What a surprise. They're a huge funder of
yeah, yeah.
research in the United States. Is
I
that true?
think that was thanks to Carolyn Best when she was still at the AUA. She's one of our friends who's been very supportive of the GBCI. She's now with PCORI. But yeah, the Department of Defense has a number of different grants that some of them are for investigators who don't have any preliminary data. They're called IDEA awards, and that's one source of funding. But honestly, the most important source of funding is usually philanthropy for this when we try to get it off the ground. We also have internal funding mechanisms here at Hopkins.
Mhm.
So Jeannie just benefited from one of those, which is the Swim Across America grants
Congratulations.
program.
Great.
So, but yeah, my vision is to use philanthropy and this other seed funding to fund projects that are going to ultimately be competitive for major peer-reviewed grants like R01 grants. I think we need the coin of the realm for a variety of different reasons. And if the quality of the research is good enough for investment, it should be good enough to potentially become one of those grants.
Um, Fantastic. So I have two questions. Please ask away. I hope this has been interesting. It's been fascinating for me. In say the last 10 years, I have a list here of at least 10 to 12 new drugs for bladder cancer, which is amazing.
Mhm.
Can you explain what in English and short, what it's not all immunotherapy drugs. So what is, we were talking about enfortumab vedotin.
Mhm.
What is that? And without going into like getting a PhD in biochemistry, what is that? And why does it work?
So it's a chemotherapy drug. So just a
Mhm.
classic chemotherapy drug. But rather than just stick it in your body and let it go everywhere, it's attached to an antibody. So an
Okay.
antibody is a protein that binds very, very tightly to another protein. It's very, very tight, and it's designed to be part of our immune system. If you've heard about COVID-19 antibodies, you've heard about antibodies. So whenever you had an at-home COVID test, you're measuring antibodies or measuring the COVID proteins, and that's how you get a positive result.
So if a patient has all the right um has the right situation and can receive this drug, this is now a new standard of care to
It's moving rapidly across all of bladder cancer.
wow.
It's so the target for this drug is not like COVID-19 protein. It's not something that's just expressed on a very small percentage of tissues. This is expressed on virtually all bladder cancers and not so much on normal tissues, although it is expressed a little bit on some tissues. So
Promising drug for
very promising drug, just basically groundbreaking.
right. How about Adstiladrin? What is that? And again, that is not an immunotherapy drug, is that correct?
It is an immunotherapy drug,
Oh,
but it's more like an immunotherapy drug like BCG. So Adstiladrin is a way of delivering a protein called interferon, which has long been used in cancer therapy. It used to be used in melanoma therapy. It used to be used in kidney cancer therapy. We even used it in bladder cancer patients for a while. But it was delivered in the blood, and it caused a lot of really serious adverse events, a lot of toxicities. It's like having a horrible case of the flu. So Colin Dinney and Bill Benedict and I, when I was still at MD Anderson, developed the idea that we would deliver this same protein in the bladder. And we deliver it in a way so that the bladder cells would make it continuously, at least for a little while. And so it's a form of gene therapy.
Great.
So we deliver it with a virus.
Wow. Wish we had more time. I have two
Yeah.
questions.
Sure.
Susan asked, why is the blue light being discontinued? Oof, I didn't know that. And what will it take? What will take its place, especially for CIS? Can you answer that?
Great question. So
And I believe Armin is doing a study on that, right?
yeah, I don't know why they're getting rid of blue light. We could talk to Armin about that in more
We will get
detail.
back to you. That's
Yeah,
like a whole conversation in and of itself. I was not aware of that. Thank you, Susan.
yeah, but
And I know
there's
that Armin has a blue light study going on, so I'll ask her.
yeah. So taking its place, I think, are going to be urine liquid biopsies.
Interesting.
Yeah.
Okay.
Stay tuned.
David, do you have any updates on BCG availability and shortages? And is there going to be limited availability, or can you say if Gemdosi study is showing promising results for as an alternative?
So what I can say is that the University of Iowa team and our team here, led by Max, have seen exceptionally good uh, results with Gemdosi outside of Bridge. We don't know what the results of Bridge are going to show, but the preliminary experience with Gemdosi very strongly suggests that it's going to be very, very potent, maybe just as potent as BCG.
A question, are patients getting Gemdosi now when if there's no BCG? Are they being substituted now, but it's not been studied yet? Is that how it's worked?
Absolutely. The other thing that's happening, we hope, is that another supplier will be on the market soon. But again, manufacturing is always tricky. It's not clear how soon there will be a huge supply of BCG available from that Tokyo strain.
David, this has been super fun. Now I have the giant question.
Yeah.
Where do you see this all going? And are you optimistic? And what can you say to patients listening in to tell them about the state of the research as it is right now?
Oh, it's super exciting because we have so many other things that we're measuring right now. We're measuring tumor DNA in the urine. We're measuring tumor DNA in the plasma. We're doing clinical trials that are based on these measurements, measuring
And that's for
microbiome.
biomarker work, right? So that
Correct.
you can be predictive.
Correct.
Maybe
Yeah.
someday your urine test will say, no, you don't need a cystoscopy.
I
Amen.
think very soon we're going to have an answer to that question.
So leave our patients, they just go through so much. We got to do better.
Well, again, we'll be talking a lot about liquid biopsies, I'm sure, over the next year, but they've really revolutionized cancer care.
Our next presentation, if you'll stick with us, everybody, is actually we're going to go in the laboratory and meet some of our lab people who are like the most darling, smart. See, we're actually going to have a little video of what the lab is and all that. I hope this is interesting to people, but it sort of shows you sort of what's going on and how it's done. Our next session will be on February 13th, and please come armed with questions. And Susan, I promise we'll get back to you about the blue light issue. We have to go to higher powers, meaning urologists, to answer that one. But this has been super fun. And David, thank you for all you do for your team. It's a long slog, but you're optimistic.
Very
Yes.
optimistic, very optimistic. And again, it only works because patients are willing to help us do our research.
Yeah. Yeah. Wow. Well, with that, thank you everyone for tuning in to our little David and Stephanie
Happy
show.
New Year, and
And just keep keep working in that lab and doing well for our people,
I
okay?
promise.
All right.
Yes.
Thank you, everybody. Good night, and we'll see you on February 13th.
Bye,
Take care.
everyone.